Hello, welcome for another session of our presentation. Now we will be on 5.2, mitosis and cytokinesis. So the key concept here is that cell divides during mitosis and cytokinesis. So when we are talking of cell division, we are actually talking about the division of the nucleus and also the division of the cytoplasm. So chromosomes normally will condense at the start of mitosis. Then chromosomes and cytokinesis will produce two genetically identical daughter cells. So perhaps when you are still a young child, you might have one time attended a birthday party or any other party, either in your home or in your neighbor's place. So maybe they were giving you bags of goodies. If you were very keen, you should have discovered that the person who stuffed the bag had to make sure that each bag had exactly the same number of erasers. If he's giving out erasers, he will ensure the number is the same in all the bags. If he is giving out candies, then even the candies must be the same number in each bag. Otherwise, some ill-mannered children or child, of course not you, would have brought a fuse if an item was missing. So in similar way, cells receive a full set of DNA, no more, no less. So they will have to work, for them to work properly, dividing the DNA is a very complicated task because the DNA is so stringy. The DNA is so stringy. By stringy, I mean it is just something which is there, like a string. So actually dividing it is very tricky and very difficult. But it is amazing that mitosis is able to divide the DNA exactly into and two nuclei and exactly without any problem. So chromosomes normally will condense at the start of mitosis. Maybe you would ask, why should they condense? So before the DNA divides, it has first of all to double itself. And it's not as easy as it may sound, because the structure of the DNA is actually, the DNA is a double-stranded molecule. The DNA is a double-stranded molecule. And apart from being double-stranded, it is also formed in a complex way that is a double helix. It is made of 4 different subunits called nucleotide. Made of 4 different subunits called nucleotides. So if you look at the structure of the DNA, you will see that it's double stranded and it's made of 4 units called nucleotides. So a chromosome is one of the long continuous threads of DNA that consists of numerous genes along with regulatory information. So we write this because it's important. A chromosome is one long continuous thread. One long continuous thread of DNA. One long continuous thread of DNA that consists of numerous genes. So it consists of numerous genes along with the regulatory information. Along with regulatory information. So when we are talking of a chromosome, it is a long continuous thread of DNA. Long continuous thread of DNA that consists of numerous genes with regulatory information. So the human body, for example, has 46 chromosomes each. Human body has 46 chromosomes. It has 46 chromosomes each. So each cell actually has 46 chromosomes. So if stretched out straight and laid end to end, the DNA is just one of your cells would be about 3 meters. The DNA in one of your cells would be, if stretched, would be about 3 meters. So imagine in one cell you have a DNA which is 3 meters or 10 feet long. That is why I'm saying it's not easy to copy the DNA. So how does this fit inside the nucleus of a microscopic cell? That should be the question you are asking yourself. How comes that such a long thread can fit in a small nucleus of a cell? The cell itself is microscopic. Not even the nucleus. You haven't talked about the nucleus. That is the cell. It's microscopic. So DNA normally wraps around proteins that help organize and condense it. DNA wrap DNA wraps around proteins. So DNA normally wraps around proteins that help organize and condense it. So it has to be organized, and it has to be organized by condensing. So during interphase, or when a cell is not dividing, DNA is loosely organized. It looks a bit like spaghetti, something like thread-like stuff, like spaghetti. That is during interphase. During mitosis, however, chromosomes are tightly condensed. Chromosomes are tightly condensed. Let's look at the figure in the next page and see how it looks like. This is how the chromosome looks like after it has been condensed. So this is what we are calling a chromosome. So this duplicated chromosome is tightly condensed. That is how it looks like when it is tightly condensed. Condensed, I told you to be condensed. It is held together by proteins. So during mitosis, the duplicated chromosomes must condense to be divided between two nuclei. So during mitosis, during mitosis, the duplicated the duplicated chromosomes must be the duplicated chromosomes must condense. It must condense to be divided between two nuclei. To be divided between two nuclei. So for it to be divided, it has to be condensed. And condensing it is very essential in terms of it dividing, the reason being that if it's not condensed, then it will not be able to divide maybe in the right order, or maybe a portion of the chromosome may be lost because it will be all over and it will be easily lost. It would be easily lost. So to maintain all the chromosomes, or to ensure that all chromosomes are present, then before division, the chromosomes must be condensed. So if chromosomes remain stingy during mitosis, they could become entangled. If chromosomes remain stringy, so if they remain stringy, if chromosomes remain stringy during mitosis, They remain stringy during mitosis. They could become entangled. They could become entangled. By entangled, I mean if this is one running like this, then the other one would maybe come and they would be disorganized and it would be difficult to separate them. So perhaps a cell gets two copies of, for example, one chromosome and no copies of the other. Maybe this can happen. So figure 2.2 shows the process that converts the chromosome from linear strand of DNA to its highly condensed form. So the key to this process is the association between the DNA and proteins. It is the association between the DNA and proteins. Let's go to the next page and see what we are meaning here. So if you look carefully, you will see here figure 2.2 chromosome structure. Chromosome structure. So this is the DNA double helix before cell division. So the DNA double helix, each continuous double-strand DNA molecule makes one chromosome. They make one chromosome. So each will make one chromosome. The DNA double helix we are saying here, each continuous double-stranded DNA molecule makes one chromosome. So here DNA and histones. So DNA wraps at regular intervals around proteins. So it will wrap itself around proteins called histones, forming chromatin. So before this, so after being coiled, we have chromatin. Chromatin interactions between parts of the histones further compacts the DNA. So even these ones will interact with each other, making it even more compact. So supercoiled DNA, so the chromatin coils more and more tightly around organizing proteins. So you can imagine if we didn't have these organizing proteins, then it would be difficult for us to have the specified genes as required. Otherwise, there would be loss of one or two bases, and this would be a problem. So condensed duplicated chromosome. Now this is a condensed one, and it has been duplicated. Can be aligned and separated by mitosis. So this one can now be aligned and be separated by mitosis. So this whole thing here, the whole of it is called the chromatin, as you can see here. Here in the middle we have what is holding here. This is the centromere. Then at the edges here we have telomere. So these telomere are important. We will be looking at how important they are as we continue discussing about nuclear division in the next videos that to come. So I hope you are getting what we are discussing and keep in touch.